I hope you enjoyed the ocean today, in a different way than normal, probably. I have actually misused my situation as a chairman to just make an advertisement here for a next meeting, which also is oriented towards people that are active in practice. The Center of Wetland Ecology is a consortium of research groups in the Netherlands working on water and wetlands. And the idea is to, from time to time, once every half year, organize a symposium for the scientists as well as the practitioners. And the first one of these will be on 14 April. You're all invited. Thank you. Next speaker of today is Merel Soons, whom I know very well. She is also in my group. And she has been very active also in the Euro-Olympics project as one of the younger scientists cooperating in this large project. And I'm very happy that she is here, and not so difficult for her as for the others because she is much closer. And she is going to talk about dispersal and recolonization by plants. That's her fascination in ecology, and I'm sure she will talk about it like that. Merel.
Well, first of all, thank you for all coming back from the ocean. When I saw the blue drinks at the beginning, I thought, oh no, this is the start of happy hour and nobody will hear my talk. But fortunately, there are people here. I have a topic that is a little bit different from what we heard before, but because we have heard a lot about the effects of climate change on the actual quality of the lakes, of streams, as habitats for organisms. And in the previous talks, people have talked about what this effect is of these changes in habitat quality for the organisms that live there. But what is also really important to realize is how these organisms arrive there. And this is especially important in restoration. So the title of my talk is Restoration in a World of Climate Change, Recolonization by Plants, and especially for the occasion, invertebrates. So there are a few ways in which climate change and restoration are linked. Climate change in aquatic ecosystems leads to changes in precipitation. Piet has talked about that and given you a lot of information about the changes in hydrology and hydromorphology that are a consequence of climate change. And he has advised all of us to take landscape scale measures to mitigate these effects. And that is actually one thing that is currently being done as mitigation effort or adaptation effort. A lot of landscape scale measures are being carried out, especially in streams, where streams are restored from deeply incised, channelized water bodies to a more meandering stream as they used to be, with gradual slopes, and streams that are able to flood again. And the idea is that this is done in part as a water retention measure, but this has also a positive effect on habitat diversity in these streams, and it can also be seen as a habitat restoration measure. So in this case, it's actually a win-win situation where adaptations for water retention or adaptations to climate change go hand in hand with the restoration efforts of stream habitats. And another thing in which restoration is linked to climate change is that we all know that climate change may lead to shifting temperatures and shifting climate envelopes. And that means that we expect that some species will migrate northwards to regions that are the same temperature as where the organisms are occurring now, but due to global warming, those regions shift northwards. And we expect that if species are not able to migrate, they may go extinct. And this also drives restoration efforts because we need habitat patches along the way for species to be able to migrate northwards. And this is another reason for restoration of habitats. So what we actually in both cases expect is that dispersal will work with the organisms. If we restore habitats in most cases we expect that the target organisms of this restoration measure, which can be Water Framework Directive indicator species, that they will follow the restoration measures and the increase in habitat quality, and that they will colonize these new sites. And also, if we create a system of stepping stones for species to migrate northwards, we actually expect them that they will be able to do that. But in order to do so, they need to disperse. And dispersal is a crucial process, of course, for species migration, but it's also crucial for species to survive regionally. If patches go extinct and new patches are created, species need to be able to disperse to keep the colonization extinction balance. And also colonization of restored sites, which is what this presentation is about. Now, if we think about the process of dispersal, there are actually quite a lot of things that we don't know, but the one thing we do know is this, that the probability of dispersal over short distances is really high, and that it decreases quite steeply with increasing distance away from the source, and that there is usually a sort of asymptotic, increasingly small number of organisms that disperse over increasingly long distances. Now, it is very difficult to estimate actual dispersal distances by plants or even of animals, because the species that disperse the furthest do so in really low numbers, and it's always really difficult to find those rare dispersal events that go far away. So what we do is we try to build models that we can test for short distance dispersal and try to extrapolate to longer dispersal distances so we get an idea over what distances species are able to disperse. This is an example for plants, and these are 4 plant species. And this is an example for dispersal by wind. This is Cirsium dissectum, this is Hypogeris radicata, Centaurea and Successia pratensis. They are 4 grassland plant species, which all have seeds that can be dispersed by wind, more or less. And what you can see is every graph is the same. There is wind speed on one horizontal axis and productivity on the other. And on the vertical axis is the distance that 95% of the seeds are, it's a 95 percentile dispersal distance, so 95% of all the seeds are dispersed less far than this, most of them much less far, and only 5% of the seeds are dispersed further. And the main point of this slide is that these distances can vary with productivity of the vegetation and with wind speed, obviously, but that they vary very much between those species, because 2 of these species have plumed seeds and they can disperse over at least a few hundred meters. But some of these species do not have plumed seeds and they disperse really not very far. It basically means that they disperse within a few meters almost all of their seeds. And another example that I can give is dispersal of seeds by waterfowl. And this is internal transport of seeds, which are consumed by waterfowl and then survive digestive tract passage and are dispersed by a bird in flight. So this is a sort of an estimate of maximum dispersal distances that you can get by waterfowl. And if you look at the distances that you can see here, they are totally different from dispersal distances by wind. We are talking about 3,000 kilometers, or for another species, 750 kilometers. But there are also species that can be eaten by waterfowl but cannot survive the digestive tract, and they are not dispersed at all because they are dispersed by 0 kilometers. So that means that we know roughly what is the shape of the dispersal kernel of species. We know that most species or most seeds of a species will always disperse over short distances, but it's really difficult to estimate the distance that a few seeds of a species can disperse. And it's also the same, this dispersal kernel is also the same for animals. And so we are stuck with a situation in which we know that seed dispersal distances can be really different for different species, and they are also different for different dispersal mechanisms. And the only thing that we know is that some species, for some dispersal mechanisms, are much better at dispersing than others. Now, we are trying to quantify what these dispersal distances can be, and the ideal situation would be that we get a database of dispersal distances for all the species that we're interested in, but we're not that far, although we are working on these dispersal mechanisms, and this is very much a research in progress. So hopefully this will at some point be the case. But what we do know is dispersal is always at some point limiting. And this means that this will have consequences for the colonization potential of restored sites. And if dispersal of plants is limited, we expect that dispersal limitation over certain distances will also affect the colonization of restored sites. And we expect this to be especially so in fragmented landscapes where restored sites or even natural sites are remaining patches in a human-dominated landscape. So what I want to talk about is what do we know about what is really happening? Is dispersal limitation really affecting the success of restoration efforts? And to give you an example of one of the few studies for which we have data on this, I'm taking an example of restored stream fragments, and I'm looking at riparian plants and benthic invertebrates. And the study that I can give this example for is a study that has been carried out in 7 restored sites, 7 restored stream sections in German mountain streams. Before they were channelized, and they are fragments in a man-dominated landscape, but after restoration their hydromorphology became more natural. I will show pictures in a moment, and the habitat heterogeneity greatly increased. And previous work has already shown that the number of aquatic and riparian habitats increased in these systems, and the question now is, can species colonize and which species do so? So this was the opening picture of my presentation. It's one of the restored streams where you see that there is a lot of variation in depth and a lot of variation in inundation depth, and also a lot of habitat heterogeneity. If you compare it to one of the embanked and channelized streams that are a reference for the situation before. This is also a picture of the reference situation before restoration. I have a few more pictures to show the situation after restoration, where you see that there are gravel banks developing, which are becoming vegetated quite rapidly. There is large wood falling into the streams, which creates additional habitats, and the wood is left there, so it is remaining part of the habitat of the stream. So we quantified the colonization by plants and benthic invertebrates 3 to 5 years after the start of the morphological restoration by vegetation surveys and invertebrate surveys, and we tried to see if this colonization was linked to the availability of source populations, by checking if the colonization was related to the presence of species in upstream unrestored sites and to the species presence in the area of, in this case, 25 by 25 kilometers surrounding the site. And we also looked if the dispersal ability of the species played a role, and we looked at lifespan, fecundity, and dispersal capacity by water and air, because these are all important dispersal traits. And if I show you the results for plants, this is the graph that shows the results. On the horizontal axis, there are 4 categories of species. And the first category of species are species that were found in the restored site and in the unrestored site. And the second category are plant species that are found only at the unrestored site, and then species only at the restored site, and finally species that were expected to arrive at the restored site because their habitat was there, but who didn't show up. And on the vertical axis, in this case, is the abundance in the surrounding area in the 25 by 25 kilometer grid. And what you can see is that the species that were present at the restored site, so in A, category A, and what is here B, indicated by a B, they were very abundant in the surroundings. And the species that were expected to colonize, but didn't, they were much less abundant in the surroundings. And what you can also see is if you look at the lifespan of the species on a categorical scale, where a lower value indicates a lower lifespan and a higher value indicates a higher lifespan, is that especially short-lived species were ones that colonized really well even if they weren't present at the unrestored site, whereas the species that were expected to colonize didn't colonize. They had a much longer lifespan. And if you look at another important dispersal or colonization trait, that would be the number of seeds that a plant produces. You see that especially species which produce really a lot of seeds colonized well, even though this particular difference was not statistically significant. And finally, if you look at the mass of seeds, so basically an estimate of seed size, you see that species with really tiny seeds, which disperse very well by wind, also by water, and especially by water birds, were very good colonizers, whereas species which has much larger seeds, which disperse less well by the three organisms, are much worse, or actually did not colonize at all. And finally, I also looked at seed dispersal in time. That is an analysis of the seed bank. And a lower value here indicates a shorter seed bank longevity, and a higher value, a higher seed bank longevity. And the plant species with the highest seed bank longevity colonized best, even if they weren't present in the unrestored section. So what does this mean? It means that plant species, there is really an effect. They do really colonize a new site more frequently if they are more abundant in the close surroundings of the restored site. And especially there is also an effect of plant life history traits. Plant species that colonize best are, as might be expected, well colonizing species. They are annuals, which produce large numbers of seeds that are small and disperse well and also survive well in the seed bank. Now, if we look at the benthic invertebrates, the first thing that we noticed was that there were no species that were present either in the unrestored site alone or in the restored site alone. There were a lot of species that were present both in the unrestored site and did colonize the restored site, and there were species that we expected to colonize and didn't. So either they were very nearby and then they colonized, or they were not nearby and then they didn't colonize. And this is also related to the abundance. The species that were present and colonized were also more abundant in the surroundings, and the species that did not colonize were much less abundant in the surroundings. Okay, finally, maybe I should go a little bit faster through the life duration, which shows no difference, and the aquatic locomotion, which showed no difference. And go to the benthic invertebrates. The species that colonize the new sites best are the species that are most abundant in the surroundings and that do not have an aerial life stage. To conclude, after restoration of hydromorphology and habitat heterogeneity, the plant species that colonize in the largest numbers are the species that are most abundant and have a life history strategy designed to quickly colonize new sites. And for the benthic invertebrates, they almost exclusively colonized when they were already present in the unrestored sections. So what does this mean? That indeed our theory that dispersal limitation may play a role is happening out there. The dispersal capacity of species and availability of seed sources and animal population sources, they affect the restoration success. And dispersal limitation does play a role in the colonization by invertebrates. And this is probably particularly true in human-dominated and fragmented landscapes. And this means that for future restoration projects, the most successful projects, they will be most successful not for all species, but only for particular species, for the species that can colonize fast and the species that are already available. And I think that that is something that we should keep in mind, because if we expect that climate change is happening and species may migrate and reach new sites, not all species are probably going to be able to do that. And this may have serious consequences for the biodiversity of species that are not able to migrate as well as others. And I think that was... Oh. Okay, I thought you said stop. Okay, so I was speeding up. Well, in that case, I have one more slide, which says, goes in a little bit more detail about this this effect of this interaction between climate change and colonization, we have been talking a lot about climate change and rising temperatures, and that of course is really important, but it has a very strong interaction with land use. And that is something that I think we should always take into account when we are looking at adaptation and mitigation measures, that there is always the landscape that has a big effect, a big impact on which species will be able to benefit by restoration measures and which species will not be able to benefit by restoration measures. And the more fragmented the landscape is, clearly the less successful colonization by many species will be. And that means that also our ideas that we especially also have in the Netherlands to have larger scale restoration projects where not only one local site is restored, but people are looking at the landscape. And for example, people are recreating connections between formerly isolated parts of the landscapes, that will really lead to better colonization of restored sites. And then, yes, I have time for the acknowledgements, because these are actually the results of a really large project. They were carried out within Eurolympics, and a lot of people contributed to the work on seed dispersal, and also a lot of people on especially the case study of these restored streams. And for those, I particularly want to mention Bob Brederveld, who is somewhere in the audience. Yes, I see him there, who carried out a lot of the data analysis on this project as my student. So that is the end.
Sorry for the misunderstanding. We have time for some questions. Any questions? Yeah, Mariet.
Yeah.
Repeat the question.
Oh, the question is, how did we know that plant species or invertebrate species were expected? We expected them based on vegetation surveys in the case of plants that were made on specific vegetation types. So after these restoration measures, certain vegetation types reappeared and could even be classified into these vegetation types. And if we compared the species abundances in these vegetation types to species abundances of a lot of typical surveys for these vegetation types, so we used books for these vegetation types from the German region where we were working, we could see which species were missing from these vegetation types that in a sort of ideal described vegetation surveys were there. And for the benthic invertebrates, we used Leitbilder. They are a German system which describes the communities of invertebrates that you can find in specific stream types. So there was, they are also based on abundances. They're a characterization of sort of the ideal situation or the least disturbed situation for this stream type.
Any more questions? So, Merel, are you a little bit optimistic about finding the data for calibrating these models? Because it's very important to find a good implementation of your model of dispersal. And with so many different mechanisms and all these factors involved, do you think that you can do that per species, or is that going to happen per species and per type of habitat? Can you comment on that a little bit?
What would be really ideal is if we could be able to estimate the dispersal distances that individual species can reach. Because usually when restoration projects are carried out, there is an idea of the target community and the species that community should consist of. But we don't have these data yet. So we need a lot more work on that, I think. But it's really important to use monitoring data after restoration projects to see which species are arriving. And I think there are also a lot more data out there which are not analyzed in this way yet, that we can use that as a test to link it to actual dispersal capacities of species to see if we are right in the distances that we are thinking they are dispersed. So we need both data, more dispersal data and the test in reality, and link the two.
Yeah. So then finally it will be possible to give even more advices for how to restore. You already gave one, like it's better to do all the restoration at once in a large landscape context rather than restoring bits and pieces one at a time. But maybe there is some more to expect if we know more about distances of species and target species for the restored sites. So looking forward to the future.
Yeah.
And thank you very much.